Hi, my name is Helen McBride, and I'm presenting a poster, Abstract 268-3334, on May 8th at the 2017 Annual DDW meeting, highlighting the functional similarity of ABP 501, Amgen, Amgen's approved biosimilar to adalimumab. This poster provides scientific justification for the extrapolation of ABP 501 clinical conclusions across all approved indications of the reference product, including the IBD indications, ulcerative colitis and Crohn's disease. First, the definition of a biosimilar. A biosimilar is defined by the FDA as a biological product that is highly similar to the reference product, notwithstanding minor differences in clinically inactive components. And there are no clinically meaningful differences from the reference product in terms of the safety, purity, and potency. The regulatory approval process for biosimilars differs from that of originator molecules in several ways. The goal here is to demonstrate similarity of the proposed biosimilar to the reference product. And one of the key differences is the importance placed on the analytical similarity of the proposed biosimilar to the reference product as it relates to both structure and function. If the results of the analytical assessment show a high degree of similarity between a biosimilar and its reference product, an abbreviated clinical program in a sensitive patient population is sufficient to establish that there are no clinically meaningful differences in the PK, efficacy, safety, and immunogenicity of the biosimilar. Similarity and efficacy can usually be established by clinical evaluation in a sensitive population of a representative indication using sensitive endpoints. ABP 501 has demonstrated similarity at all levels of the characterization process, from the structure and functional assessments through clinical evaluations in two indications, RA and psoriasis. Because the clinical program for a biosimilar is abbreviated, not all clinical indications for which the reference product is approved need to undergo clinical evaluation. Thus, it is important to demonstrate that a proposed biosimilar shows similar activity in all key mechanisms of action, MOAs, across the indications approved for the reference product, and that a high degree of similarity is demonstrated in all of these. The primary MOA of adalimumab and ABP 501, Amgen, is the inhibition of soluble TNF binding and inhibition of downstream signaling of TNF-alpha. In addition to that primary mechanism, additional MOAs mediated through membrane-bound TNF may play a role in IBD indications. This poster presents data demonstrating that ABP 501 is highly similar to adalimumab based on the results of assays that represent the established and potential MOAs of adalimumab, including soluble TNF binding and signaling, membrane TNF binding, reverse signaling, and the effector functions, ADCC and CDC. These data, when combined with the previously reported similarity demonstrated in physical-chemical properties, pharmacokinetics, efficacy, safety, and immunogenicity in sensitive patient populations, provide the scientific basis for the use of Amgen across all indications approved for the reference product, including the IBD indications of ulcerative colitis and Crohn's disease. Please drop by the poster and see the data for yourself.